A good morning or afternoon to you, depending on where you are in the world. Thank you for joining us today. I'm Nick Hutchinson. I volunteer at the Antibody Society, and I will be hosting our event today. Today's webinar is one in a new series designed to inform and educate our members and the broader scientific community about topics related to the commercialization of antibody therapeutics. Our expert panelists are Drs. Martin Welschof, Christine Power, and Björn Cochlovius, and Dr. Tom Burt will be asking the questions. They will be discussing developing a portfolio of first-in-class immune-oncology antibodies. Please note, this webinar is being recorded, and if you have questions, please do add them to the Q&A box in the viewer. Panelists will answer those at the end of the initial discussion. So without further ado, let's get started, and I'll hand you over to Dr. Tom Burt to kick off this fireside chat.
Many thanks, Nick, and glad to be here. As usual, we have the initial technical troubles, which hopefully we've managed to overcome. It's great to be here. My name's Tom Burt, and I actually have a background in bioprocessing, but have moved on to the investment world, where we look to invest and support companies, innovative biotech companies, much like those represented by the panelists today. I think it'd be useful to start by just hearing some of the backgrounds of the panelists today, to get a better sense of where they are and where they come from. So Martin, I don't know if you want to start, and then Christine and Bjorn, just a brief intro would be really helpful.
Yes, thank you, Tom. Happy to start. So my name is Martin Welschof. I'm the CEO of BioInvent. And my background is I'm a scientist by training, but I'm now in the life science industry for roughly 25 plus years, having worked in the US, in several parts of Europe, and since now August 2018 at BioInvent. Nice to be here.
Thanks, Martin. My name's Christine Power. My name's Christine Power, and I'm currently working as a consultant at my own company, which is Christine Power Consulting, based in the Lake Geneva area of France. So I've worked in antibody discovery and development since about 2006, so about 15 years. First at Merck Serono in Geneva, followed by Arsanis in Vienna, where I was working on the discovery of antibodies for infectious diseases. And then most recently at GSK in Stevenage, UK, where I was head of antibody technologies. So thank you for inviting me, and I look forward to this webinar.
Thanks, Christine.
Hello, good evening, good afternoon. My name is I'm a molecular biologist by in the antibody world, more or less since the beginning of my career. Academic-wise, I was working with research center and Weizmann Institute in Israel in 2002 in various positions in small biotech companies in Scandinavia, for example. Also worked in all good old times. And later then I joined, I was with Japanese Otsuka and the American in Singapore, Europe again, consultant, Christine. I'm sitting, I'm in 3 companies. I'm chairman, including Karolinska Development, arm of the Karolinska. And I'm founder of a new CEO of a company in Heidelberg, and since last week I'm a partner in a new life science VC in Hong Kong. Looking forward to discuss.
Well, thank you all. It's incredibly relevant backgrounds and a deep experience here. I mean, obviously this is an event hosted by the Antibody Society, but the talk today is more specifically on developing immune-oncology antibodies and so developing a first-in-class IO portfolio. Obviously, Martin, you we know BioInvent is predominantly focused on immuno-modulatory antibodies. So I think it'd be very fitting to explore a bit about the history around IO, rather than saying immuno-oncology every time. I'll shorten it to IO. I'm sure everybody knows the acronym. And so, yeah, really just to find out a bit more about the history of IO, the impact it's had on cancer treatment. And so really be useful for each of the panelists to just briefly summarize where we are with IO approaches, how we got here, and more broadly, how the potential of such approaches in cancer going forward. So again, Martin, I don't know if you want to kick off on that sort of aspect.
Yeah, happy to do that. So obviously the big kickoff of IO was the introduction of pembrolizumab and ipilimumab. Those were the first, and then followed by a couple of others, antibodies that really were targeted against new structures, which are basically inhibitors, so providing activating signals as well as inhibitory signals on T cells, and CTLA-4, PD-1 just being one of those. And those antibodies, so Keytruda, for instance, obviously had quite some success. But when you really look at the patient population that is treated, so you can see that still the vast majority is not getting the benefit of those new IO compounds. So I think it's around 10 to 15% of a response rate. So there's still a lot of space. And then more recently, obviously, a lot of companies moved into the field in order to come up with new structures that could complement or maybe even go on top of those drugs that were introduced first.
I see. So, I mean, it's going back to the first old days of Keytruda, and obviously ever since then we've we've seen continued expansion of the use of PD-1 and checkpoint inhibitors going beyond that. I mean, Christine, Bjorn, I mean, any further detail about how you view this, the initial days of IO and how we got here?
Yes, I just wanted to add that most recently there has been quite a lot of development in the field of bispecific antibodies, so particularly targeting CD3 on one arm of the antibody. And then, for example, the initial bispecific antibody was a BiTE, which was with one CD3 arm and one CD19. And now many companies are looking to develop these kind of bispecific targeting antibodies, not only with CD3, but with other second arms against tumor-specific antibodies. So this has really quite vastly opened up the targeting space in the immuno-oncology field. And yeah, it would be interesting over the next few years to see how many of these molecules which are currently in clinical trials actually progress to the market.
Absolutely. I mean, obviously, since Blincyto, I mean, Bjorn, from your angle, what's is notable about the around IO approaches in cancer?
Yeah, I mean, I'm personally very happy and satisfied. IO is becoming, if you want to call it like that, if the times when we were young, you probably remember the same, that in those were always not really in immuno-oncology. When we told immuno-oncology, we believe that the immune system can beat cancer. To help it a little bit, people always said, no, this will work, it's too weak. And skepticism toward oncology for a very long time in the academic field, but also took a long time to be overcome. And this has overcome only what Chris about the bispecific, for example, which is now coming to market 20 with these kind of bispecific academic environment. This just shows immuno-oncology to throughout. That's a very interesting thing for. You have a lot of possibilities now which are opening up, and I'm sure that to detail. Not only having on effectors, also at the microenvironment and all these other things. And now we have PD1, PD-L1 in the picture, away from this purely to and are going into opening up new fields where we with other aspects in this field, well, that are tumor target. Very important topic also tonight, I'm sure.
I mean, that's very helpful, Bjorn. I think it's it's encouraging to hear, and it's true to hear that there's almost an embarrassment of riches of targets to go after. Broadly speaking, both in IO and then obviously looking broader into antibodies themselves. I mean, I suppose from our angle as investors, we're always very keen on equity stories that have both breadth and depth in the pipeline, and by that I mean obviously a clinical stage developmental pipeline products that you can hang a valuation on, but also the platform from where those assets originally are discovered. I know, Martin, in BioInvent, you have the first discovery platform, but that mechanism of being able to refresh the pipeline, but also having those early stage or late stage clinical assets from which investors can say, well, this company is going to meet these milestones, valuation progression is going to be this, X and Y. And so you need both, if you like. And I suppose a question, first off, for both Bjorn and Christine is, when you're advising your clients, where do you strike the balance between those pipeline assets and the platform? Obviously, the pipeline is critical to driving interest, both from investor standpoint, but also from the clinic. But to get there, you obviously have to focus on the platform, and that is key. So I just wanted to be interested to know how you advise your clients on striking that balance between platform and products.
Christine, you want to?
Yeah, shall I go first? Yeah, shall I go first? Well, I think it's quite important, in terms of, for example, if you want to develop a bispecific antibody, so for example, for T cell redirection or for NK cell redirection, I think the first thing is to make sure that your targets are pretty well validated before you go in. And it's also extremely important to ensure that you've done, a sufficient look at the market potential of these antibodies in terms of the number of patients, and whether you're going to have to do patient stratification, whether you need to have the particular diagnostic assays in place so that you can select a population where you are going to actually see a response. And so I think they are probably, the main criteria that you need to look at before you start the actual clinical development.
I see. Thank you. Bjorn?
Yeah, thank you. Christina, very right. So when I talk to clients on, balancing, working more with the platform or pipeline, I say to them that they should first show and validate their platform. So that's potential collaborations in that they can showcase their. So until they have really data that shows doing a good foundation, and we are of course and that's too late. Those should pipeline projects. That this should be rather they should start thinking about it, not only pipeline. That's not only important for investors, important to showcase themselves and that they have not only this, we can do projects, but they have also this beyond that own platform to thinking about projects that have that should have an edge on the market. That makes them for trade sale folks in big pharma companies. They then really only able to work perfectly with their brilliant platform, but their capabilities of innovation. I believe that's very important.
I see. And so, I mean, it sounds very much rings true that, I mean, validating the platform is key before, progressing. I mean, having said that, I mean, obviously you're all working in the field of immunology, which in itself is rapidly progressing, and of which we understand is inherently very complex. I mean, Martin, I mentioned the first discovery platform where very effective method of identifying potential IO targets. I mean, I was wondering when you and BioInvent look at this problem. I mean, do you have the confidence in your platform, but also the underlying immunological principles at work here to go after novel, new de novo targets, or do you like waiting for the science to have evolved slightly? I mean, taking CD47, as thinking that all right, that's fairly established now. Should we go and try and find a target in that sort of that space? I'd just be interested to know how you look at it. I mean, are you purely focused on very novel, yet unproven targets, or do you look for some risk mitigation and try and go after more established pathways?
Yeah, happy to comment on that. So before I do so, maybe just very briefly, connecting to what Björn said, I think that's quite important. Not only to generate confidence for yourself, but also for later partnering or maybe even larger partnering events. So we did the same. So BioInvent obviously validated the platform significantly. So more recently with Pfizer, but also with Daiichi, Takeda, Mitsubishi, Bayer. So they all have targets and antibodies that we have discovered with first in their development pipe. So I think that's important. And then regarding what we're going after, so first of all, maybe I have to explain very briefly, not too extensively, the platform. So first of all, so we screen directly on patient tissue. So our current focus is oncology. So then it will be on cancer tissue. And we have set up a very close collaboration with the local hospital in Lund, where the company is based, such that we receive high quality, fresh tissue on a regular basis. And then we use those materials directly in the screen. So we don't do any modifications. We don't take it in culture. So we receive it from the hospital, and then 10, 15 minutes later, we start the screening. And then we identified first specific antibodies to the cells of interest or to the material of interest. And before we even check what those antibodies are binding to, we subject those antibodies to vigorous functional screening. So we have a number of animal models set up in-house that those antibodies go through. And then we only focus on those that we have really seen superior therapeutic effects in a handful of different animal models. And then we identify the target. So the selection is purely function driven. And what we get is basically 3 different categories. So we see, of course, completely new stuff. We see the PD-1s, the PD-L1s, CTLA-4s, etc., OX40, name them, GITR. So we have all our own versions in-house that we have selected with that method. And then we get antibodies that, or targets and antibodies that are known, so where you know some biology around the target, but maybe not in the context of IO. So this is our program BI-1808 and BI-1910, where we're targeting TNF receptor 2. that we identified on ovarian cancer tissue, where it was highly upregulated on the T regulatory cells in the tumor microenvironment only. And what we have in our portfolio is a mix. So we have first in class. So most of it is first in class, but we also have one program where we push ahead with our own proprietary anti-CTLA-4, which we do in collaboration with Transgene in the context of an oncolytic virus. So it's a mix, and with the platform we see the mix. And where I have a preference is really targets where some biology is known, because then you don't fish completely in the dark, but where the context of IO is new. So, for instance, the TNF receptor 2 program.
I see. And so, I mean, building on that, I mean, I think Christine mentioned, a lot of interest initially from biospecifics, more considering the tumor microenvironment, these more novel angles in IO. I mean, obviously, I'm not saying we've been here before, but we have seen other IO targets that have shown initial promise very early on. I mean, I don't want to name names, but A2A, IDO, for example. I mean, where a lot of promise early on, and then perhaps translating that into the clinic at the moment has not fully been realised yet. I'd be interested, both Christine and Björn and Martin, how you go about, specifically for IO targets or novel IO targets, how you build the story around. A novel target which BioInvent may be progressing, and how that is different, if you like, and given there is the complexity we're talking of here in immunology, what obviously we'll move that into the clinic, and that's where the proof is in the pudding. But pre that, it'd be interesting to know how you differentiate these specific targets.
Yeah, I can briefly start from the BioInvent perspective. So what I didn't mention is that we have a very strong immunologist group in the company. So we have 10 to 15 really high-class immunologists. So I think that's very important, because once you get a new target, you really have to understand the biology and the context in immunology. And then another thing, what we do, which I didn't mention, so we always follow through 2 tracks. So we have a so-called mouse track, and we have the human track. The human track is obviously the human candidate that eventually then will go into clinical development. But we also always develop very closely match mouse surrogates, such that we can also test it completely in the mouse system. That means not in a transgenic system, but really in a mouse that has a normal immune system. And obviously, you cannot translate that one-to-one into humans, that's clear. But at least we try to do as much as we can preclinically then before we move it into the clinic. Mm-hmm. And those would be the approaches. And I think besides the platform that I have described, BioInvent has built over the years a very strong expertise in immunology, which I think is key.
I see. Honestly, so it's really building that preclinical package, obviously ahead of clinical trials. I mean, Christine, Björn, I mean, does that ring true to you both? I mean, when you're advising companies, and perhaps you can talk about this, beyond IO companies. I mean, how do you build interest in this novel target, which has as yet perhaps not moved into the clinic? About building that preclinical package to convince both investors, but also clinicians and potential partners.
Yeah, I think if I could just step in, I think one of the most important things is that once you've actually, done all of your validation of the molecule, one thing that's often not really spoken about very much is that you have to make sure that, whether it's a monoclonal antibody, bispecific, or, ligand trap type of antibody, bispecific antibody, one of the things that you really have to do is to make sure that your product is actually developable. So, it's not going to fall apart. For example, if it's a bispecific, it's not going to be degraded. And it has all of the, necessary characteristics that would facilitate the actual manufacture of the molecule. So it's not really just a question of having the fantastic functional activity. You really need to make sure the molecule is developable from a CMC manufacturing point of view.
That's a very good point, and I we'll touch on manufacturing later, and coming from the manufacturing world, I think it's historically been very overlooked, and something, these things can come back later on and be very disruptive, and obviously by that point you've invested a lot of capital to get to that point. So very salient point there, Christine. And Bjorn, I mean, I don't know if you've any comments on how you build that preclinical package, specifically for IO or antibody.
Yeah, I mean, one part of your question, of your original, I believe I would like to address, where you ask about new targets. So what do you do when you have a new? It's very important. I'm here to know how on being your new target. You don't want to have any bad surprises when you're working with that in organisms. So I have to have invest a couple of years to work on. Although it might be painful these years, of course, perspective, but I believe it will do that. I mean, preclinical development. The other points have been addressed, specificity, etc. What I believe is important, and it becomes more and more important in the field of, especially, is that the possibility to find other therapeutic agents. Because as PD, big hope, but we learn more and more in the clinic that probably it's a therapy that needs a combination of other therapies. So I think that needs to first address this relatively early in the development, because it shows the possibility. If you see good combination, shows that there is not only monotherapy, and that sort of later again on your potential big pharma.
Yeah, I mean, Bjorn, it's a perfect segue into discussing the monotherapy combo discussion in IO. I mean, Martin, I know I think January this year, BioInvent reported very strong proof of concept data for 1206, which is your novel Fc gamma mAb, which is being used in conjunction with or in combo with rituximab. I think from memory you're talking 6 out of 9 partial responses and 2 durable complete responses. So very impressive data for a refractory population that you were testing this in. Obviously, the strategy around 1206 is as to sort of bind, to block macrophage binding, and so by definition, it is designed to be in use in combo. But I mean, obviously, a lot of the big debate in the IO space is combo versus monotherapy activity. I wonder where you stand on that sort of dogma of combo versus monotherapy. How important is it for you to show some degree of monotherapy activity with your agents? Obviously, in the understanding that some of the agents that you're developing are being designed to be purely working in combo. But I just wonder where you stand on that discussion.
Mm-hmm. Yeah, so I think it generates great comfort if you can see single agent activity. So obviously what we will do, and talking very briefly about the lead program 1206, where we try to develop this antibody in 2 different settings. One is in non-Hodgkin lymphoma, where Fc gamma RIIb, which is a target, is expressed on the cancerous or tumoral B cell. Whereas in the solid cancer setting, we're targeting Fc gamma RIIb when it's expressed on the innate immune cells of the tumor microenvironment, so purely modulatory. Also in this case, when we looked at the non-Hodgkin lymphoma case and we characterized the mode of action, we saw two activities. So we have single agent activity because 1206 is an IgG1, so can attract and bind to macrophages. But the main position is really that by blocking Fc gamma RIIb, you block one of the major resistance mechanism of anti-CD20 based therapy. That's the main thing. And so the positioning is clearly as a combo. And as I said, so it always gives comfort when you see single agent activity. But my conviction is that at the end, when you look at cancer therapy, it will be combination. So, even with strong assets as anti-PD-1, anti-PD-L1, anti-CTLA-4, at the end, there will be combination. And I am 100% agreement with Björn. And that's also what we do preclinically already, that we already test various combinations in a number of different animal models to see what are the effects. Do we see any synergies? And of course, even more importantly, the toxicity aspect, because if you have something which is, induces a toxicity burden, and then you want to pair this up, just as an example, with ipilimumab, which also has its toxicity package, then you will be in nowhere land. So I think it's quite important to have that as part of your development strategy, that you can combine. And that's also what we do, for instance, with our TNF receptor 2 program. We have 2 arms in the clinic. So one is single agent activity testing, where we develop it as a single agent. And next year we'll start enrolling in combination with pembrolizumab. So I think it's good to see single agent activity, because it also helps you in the partnering process, also later in the development process, because then nobody can say, well, look, this is just, responding. So it's really that you have activity, but at the end, my conviction is it will be combination for most of the drugs.
Mm-hmm. That's clear. I'm very well understood. I mean, Björn, Christine, is that your view here? That again, we are talking about combo here, and so if you start from that preset initially, that it will be developed for combo. So you're not overly concerned about single agent activity.
Yeah, I think that the combo is the direction that we're really going in at the moment. As long as you can actually show an effect of the combination, and sometimes administering 2 single antibodies, or in the format of a bispecific. In bispecific format, when you choose the combination that you're going to work with, the geometry of the molecule is particularly important to make sure you engage both targets correctly. And I think this is particularly important for, if you want to have an antibody bispecific, which is recruiting cytotoxic T cells or NK cells. And sometimes, indeed, with a actual bispecific format, you may actually see an activity which you don't see with the combination of the individual antibodies. So it's certainly a direction that we need to move in.
Mm-hmm. Understood. Well, I mean, in terms of Björn, I don't know if you want to.
No, I just wanted to add one sentence. Sorry for that. So in general, it makes sense to first show monotherapy efficacy of the drug, whatever, and then go from there. So you don't want to just because it didn't perform properly as a therapeutic agent before. If you see, for example, suboptimal, if you see toxicities, then maybe. And at the end, I agree with what Martin said. Combination will be many things in oncology. To the point of sometimes you have on top combined two drugs, and your new drug is made certain you can maybe in the combination also reduce the drug. You can use a potential side, and if the combination has a lot of potential, 100%. So that's the future. That 10 or 12 years from now, you will get anything prescribed oncologist or used in the hospital anymore.
I see. Okay.
Tom, just to interrupt, sorry, Björn, we are having, some of the audience members are having a bit of trouble hearing you. I don't know if it's possible that you might have a headset that you could use. It will be useful if that is at all possible. And apologies to the audience members that are having trouble. Okay, sorry, Tom, didn't mean to interrupt.
No problem, Nick. It's more technical challenges, as we know. Technical challenges exist in tech and in biotech. It's just the way of the world. I mean, just talking on combos, Martin. I mean, obviously, you look at BioInvent's pipeline, and I mean, you're combining novel IO targets with anti-CD20 mAbs, checkpoint inhibitors, also with oncolytic viruses. So it's quite a breadth you have in what you're exploring in combo strategy. I mean, how do we look at that? Is that you having a specific rationale here that in this case we think there's a clear underlying immunological reason why combining this with an oncolytic virus would make sense, or an anti-CD20 mAb, or is it more exploratory at this point? Is it, well, we know we have an agent that is provoking this specific immune response, therefore let's see what works. I mean, I'm sure it's a lot more scientific than that, but it'd be helpful to see what guides you in your combo strategy.
Yeah, so what I can say in summary, it's purely data driven. And I can maybe give you one specific example. So you mentioned BT001, which is our proprietary anti-CTLA-4 combined with an oncolytic virus. And so when we identified also with the first platform, our anti-CTLA-4, we found an antibody that was different to ipilimumab, which is the current one, which is used in the clinic. In that sense, it binds to a different epitope. And then when we compared it and characterized it carefully head to head, we found out that it has the same blocking capacity as ipilimumab, but a much stronger Treg depletion activity. And then, of course, we were thinking, well, if it's a more potent Treg depleter, maybe there might be more toxicity issues around this. So then we thought, okay, what if we can put it into a kind of a Trojan horse? So, the oncolytic virus platform of transgene is a vaccinia virus that can take big payloads. So basically, we cloned in our anti-CTLA-4 into the oncolytic virus, into the oncolytic vaccinia virus. And then what we do is intratumoral infection. And what we saw very nicely preclinically, and we just had another poster 2 weeks ago on SITC, is that we have a very, very high expression of anti-CTLA-4 in the solid tumor environment only, and a very low systemic exposure. So in this case, it really paid off, the strategy that we went for. And based on that, then we initiated also clinical development that is running since beginning of this year. And we hope to provide the first data on that during first half of next year. So that's just an example. And the same is true for the combination with 1206 with rituximab, where we did a lot of preclinical work, and we did not only check rituximab, we checked also a number of other anti-CD20 antibodies, and there was a clear reason why we moved ahead, how we moved ahead, and what we have in the clinic now. The combination with pembrolizumab, where we really did all this testing, because we have all these antibodies in-house, the different PD-1s, CTLA-4s, etc., such that we can really match very carefully and really see where we can enhance and get the best effect, at least preclinically, and that we move them into the clinic.
I see. So, I mean, perhaps we can so effectively, it's all data driven here. This is on understanding the underlying immunological mechanisms. I mean, the advantages that we're speaking here, obviously, of the combinations being derived often encourages quite a lot of partnership. Now, I know, Martin, in BI, you have partners with Pfizer, Transgen, Daiichi. And I mean, the importance of partnerships is obviously relevant, not only just in IO, Christine and Bjorn, any antibody company having that, that sort of outside interest, not least, but obviously some degree of validation to the pipeline or to the platform. But I was wondering, Christine, what other advantages when you're advising your clients about potential partnerships? I mean, what is the front and center view? Is it about proving the science, is it the validation? I mean, what other things are of interest to companies when they're seeking a partnership for one of their key assets?
Well, I think when you're seeking a partnership, it's important to think about what you can actually do yourself, and what the partner, what expertise the partner can actually provide. And so, for example, if you're a rather small company, there's a limit to what you can do in terms of the clinical development, and also in terms of the funding, the resources that you need for the very expensive clinical trials and so on. And so that's what you're really looking for, from a small startup point of view. You want a partner who can actually provide the expertise and resource that you just do not have either the capability of doing or the financial resources to do it.
Understood. So they're bringing more to the party than just an agent. It is what else? The clinical development, perhaps. I don't know in case with Pfizer, Martin, if they're bringing their own PD-1. I mean, so it's those assets, it's those resources and capabilities. I mean, Bjorn, is that play into what you see when you're advising clients? Is you've got to look beyond just the asset. It is what else do they bring to the party? Oh, Bjorn, I think we can't hear you at all now. I think
he's on mute.
Oh, he's on mute.
Maybe while he's unmuting, I can maybe comment a little bit on the partnering strategy that we have followed through at BioInvent. So there are different phases, basically. So the partnerships that you mentioned, Tom, that were R&D collaborations, where we basically used our technical capabilities to generate targets and antibodies for those companies. And then since a couple of years now, we have moved ahead with our own portfolio. So we build partnerships around that. And then, for instance, we really look more for partners that can help us in our clinical development. So, for instance, for the 1206 program and 1808 program, we have supply agreements with Merck, such that we get access to pembrolizumab for free. Mm-hmm. We also have done our first geography-focused partnering around lead asset with CASI Pharmaceuticals. That was commercially attractive, but not only that, because they become now our partner in clinical development, because they will develop jointly 1206 together with us under our international worldwide clinical development strategy, but obviously give us access to interesting patients in China. So that's another aspect. So absolutely, Christine, as you said, so there's funding, there's validation, but there's also useful things that you can obtain in a partnership. And going forward, we'll probably more partner around the portfolio, but very specific, and probably the way going forward is geography-focused partnerships, such that we retain enough upside for later stages when hopefully we have good data. But we also, always have the possibility to partner around the platform itself, because the discovery engine is still running. And I think what I also should mention at this stage is that BioInvent is relatively integrated. So we not only have the discovery piece and development, preclinical as well as clinical, but we also do manufacturing. So we can produce our own cell lines, can produce up to 1,000 liter GMP, which is very helpful because like this, we can go very rapidly from discovery into clinical development, because while we see already there's an interesting program building, we generate the cell line one floor up, and then at the right time point, then we can kick off the manufacturing process. That was quite helpful because within 3 years, we're building up from one program in the clinic to, by the end of this year, 5 programs in the clinic.
I see. And I mean, this is BioInvent is a very established company now. It's perhaps gone through that early phase of growth. But I mean, perhaps Christine and Bjorn, I mean, when you're advising your clients that maybe have built a very effective preclinical package, they may have sought partnership. They have de-risked a lot of the fundamental underlying immunology, and they're moving into the clinical stage. That in itself is associated with a shift in strategy. How do you guide clients when they're making that transition from preclinical to a clinical research organization stage biotech? I mean, you mentioned in your previous answer, Christine, about getting involvement from the from the partners around clinical trial design or clinical development planning. I was just wondering any other issues that a biotech should be aware of as it makes that move from preclinical to clinical. So I'd ask Christine first.
Yeah, I think it's also important to understand what the strategy is. In my case, I'm working mostly with startups. Mm-hmm. What they actually want. Do they want to be acquired by big pharma, for example, or big biotech? Or, how much, do they want to do themselves in terms of getting the funding for phase 1, phase 2 clinical trials, for example? Because, that's one of the, the difficulties, really. You have to have a very, very solid data package, and also, nice, convincing results. And, depending on what you have, this is going to influence the stage at which you're going to either go into a partnership with a company, or if you just basically, as I said, if the goal is to actually be acquired. So, this depends very much from startup to startup, what their actual ambitions are.
I see. So it's a case-specific basis.
Yeah.
Björn, anything to add on that in terms of advising clients on that sort of step change from preclinical to clinical?
Tom, we unfortunately we appear to continue to have some trouble contacting Björn, so we'll be trying to fix that.
It's again, we're a few technical hurdles, but nothing that couldn't be resolved, much like we see in drug discovery. I mean, Martin, if I can just ask about, obviously you've moved into the clinic now. You're, obviously in BioInvent, you various number of clinical programs. I mean, when you've had clinical programs before, I'm not saying this is the case in BioInvent, but I don't know if in previous companies where perhaps the clinical data is not read out as first hoped. Obviously you've got biotechs don't have endless resource. How do you go about rationalizing decision making based on clinical data? Is it purely guided by that snapshot of clinical data from the latest trial, or are there other factors at play which may lead you to deprioritize a program? Or is it purely the clinical data, as you mentioned with the preclinical setting, or is there other factors?
So in principle, can be both. So obviously, first, just to address your first point, that's why we have a portfolio, because obviously we all know that very, very good preclinical validation characterization doesn't guarantee clinical success. Unfortunately, it doesn't work that way. And that's why it was so important for BioInvent to push ahead with a portfolio. And as I said earlier, sometimes I think we're still also running the discovery engine. So we're still identifying potentially new, interesting targets and antibodies. And then, as I said, it's twofold. So obviously data is important. So if you can't translate what you have seen preclinically, clinically, then obviously it doesn't make sense. And then, of course, what you also have to do is really to monitor what the competition is doing. So let's say you have good data, but then suddenly somebody is bypassing on the right or on the left with a compound that is so much better, because at the end, of course, you have to measure yourself with the standard of care which is out there. Sure. So that could also cause the stop of a program if you see it doesn't have commercial value anymore, because that's the combination at the end. So you have to have good clinical data, you have to see efficacy, you have to have good tolerability, and then of course it has to be a commercially viable product. You could always argue that as long as you target high unmet needs and you really help patients, that there should be also commercial value. But of course, this has always to be seen in the light of competition. And if there's competing products that might look more tolerable, maybe a little bit more efficacious, then you might consider this and say, okay, fine, maybe that program doesn't have a future anymore. And that's why I think it's very important, if you can, to have a portfolio, because then you can basically move to the other program and put more emphasis on another program. And that's also obviously how it will work. So let's say suddenly you see very strong data on one program, you might focus your resource and say, oh, now we push ahead with that one mainly, and the other one go a little bit on the back burner.
You're preaching to the choir there when you talk about multiple programs. Obviously, it is. As we all know very well, biotech is a risky game, and so having that multiple shots on goal is risk mitigation in itself. So very, very clear. I mean, one thing you didn't mention. I mean, I don't know whether this is a reason. Does manufacturing ever come into play here? I mean, Christine mentioned earlier on about sometimes getting that work done early on. That have you actually got a product here that can be fabricated and made on a commercial scale? I mean, I don't know how often that comes into the thinking. Obviously, you'd hope by that point that you have a product that can be made. But it'd be interesting to hear, all of your views on the benefits of owning that manufacturing. Obviously, I'm sure a lot of attendees of this call, get it drilled into them about the processes, the products, and the importance of really understanding the manufacture of these drugs early stages. So Martin and Christine and Bjorn, please jump in if you're back and able to. I mean, how important to you is controlling manufacturing? And I think part of that question is, when is the best time to invest in manufacturing, given obviously the expense of fixed assets?
I think it's very important. So I think for me, that was actually one of the key aspects why I joined BioInvent, that we had our own manufacturing capabilities. For various reasons. So first of all, you can control much better timelines and costs. And it's a very important aspect of development, as Christine already outlined. So obviously you need to see that you have a compound that is stable, that works, that has a good shelf life, etc., all those aspects. Plus, you need, of course, a good cell line in our case that has a good yield, that generates a commercially viable product. Because if you, let's say, have something which produce at 2 grams per liter, forget about it. You need to achieve 4 to 5 grams. So I think that's something that we look at very early. And obviously we have the luxury that we have it in-house, because that of course gives us a very cost efficient and very manageable way of implementing this. I have run companies before where I didn't have that in-house. And then of course it's a little bit more challenging, because then when do you spend the time to really make a cell line first and then also start producing material? And that is mainly also data driven, but some things probably you have to kick off at some time point, and then it's an opportunity cost.
Absolutely. I mean, so Christine, any comment, and Bjorn, any comment on when you would advise potential, your clients, but when to invest in manufacturing? I mean, obviously, a preclinical biotech wanting to construct a 1,000-liter process suite and all the added extras. I mean, perhaps doesn't seem a judicious use of finance. So I don't know. I mean, any thoughts about how you advise your clients on that front? Oh, Christine, I think you're on mute.
Yeah, from a startup perspective, I mean, it's really often not feasible for a startup to, do their own, scale up and manufacturing. So I would definitely advise to identify a partner for, making the cell line, and actually then going to scale up to produce the API. Now, and also, you know, enough material for the phase 1 and safety and tox studies. But, you know, I think that before they engage in that, you know, because that's a very, very costly thing to do for many startups, what they have to be sure of is that the antibody candidate that they want to develop the stable cell line for is actually, you know, they've gone through not only the functional validation, but they've done as much as they can to make sure that this antibody is developable. And so they've addressed all of the issues like stability, solubility, potential immunogenicity, and so on. And, you know, not only to make sure that they have a candidate, but possibly to have a backup just in case something goes wrong during the cell line developing and manufacturing process. These kind of things can happen. And so it's always a good idea to have a backup candidate as well.
I see. I mean, I think we're very, very helpful, Christine. I mean, I think we're coming up to the top of the hour. So I did have a question on finance. But you know, I mean, coming from the financial world, I don't know how relevant it is. I mean, obviously, we're all there trying to get finance from our respective shareholders, and obviously that is data driven in itself. Martin, I know you've recently closed a transformational financing with Redmile, and you know, it seems quite an assured path towards more of a US capital markets angle, and so again, getting that continuous access to finance, which, you know, developing these leading edge medicines it always requires. But I suppose my final question would be, we all like to end with a bit of crystal ball gazing and predictions. Just about a bit of fun. I mean, in a way, we see multiple developments going on in the oncology field. We talked about novel IO targets today. Obviously, we have cell therapy. We have a lot of activity in precision oncology. I mean, looking at your portfolio, Martin, we know that if 1206 lives up to the promise, then, you know, it kind of offers the equivalent clinical benefit to a CAR T approach without the expense of production. And so, given all these very fast-moving developments, it'd be very interesting to hear your view on. Take a look at that crystal ball and where you see some of the exciting developments coming in the near term. And perhaps I'll direct that to Björn to stand off, but I hope he's back with sound. Clearly.
Oh. Excellent. So I believe one point that's now a couple of times is the combination of that will come for sure, and so this is not really a crystal ball. Another thing that on the antibody world in the future is I expect getting next couple of years from new efforts to target antigens, and here I tumor tissue, but also on other target cells, as a combination from tumor therapy that interacts tumor suppressive functions of, you know, this is some but I believe there's so haven't been unlocked yet. So there's a directly or indirectly, but there's still a huge universe of antibodies in immuno that we still haven't touched.
Okay, very interesting. Christine, I don't know if you'd get your crystal ball out.
Yeah, so where I agree with Björn's comments. I think obviously we need to look at the target space. One of the areas which is underserved by antibodies, if at all, at the moment, is intracellular targets. So I think with my crystal ball, I think that we're actually going to see the potential for antibodies which are able to internalize and, for example, again, I'm a bit bispecific focused here, but can carry a cargo into the cell, much like an antibody drug conjugate, for example, but this time deliver intracellular antibodies for, targeting many of the intracellular antigens which are specifically expressed in tumor cells. So this is an area which I think is going to develop in the near future.
Yeah, very interesting. And Martin, beyond BioInvent becoming the next gen mab, where's your crystal ball pointing you?
Yeah, my crystal ball is pointing me to the tumor microenvironment, a little bit what also Björn already has said. So I think what has been done quite significantly already is targeting the adaptive arm. So a lot of Treg targeting, inhibiting or activating. But if you look at the innate immune system and specifically the dendritic cells, tumor-associated myeloid cells, because if you look at the tumor microenvironment, the bulk is tumor-associated myeloid cells. And I think there will be also a lot of interesting targets, inhibitory or activating, that you could use in order to modulate the tumor microenvironment, which is actually quite important for the persistence and the spreading of cancer.
Sure. Okay. Well, Nick, I think we might have come up for the top of the hour. I mean, I think it's been a very rapid sort of progression through all facets of developing a biotech, from target identification, the platform, to preclinical data packages, clinical development, manufacturing. So it's covered a lot in an hour, and obviously we've not done that in a huge amount of detail on each topic, but I think it's been incredibly useful and insightful advice from everybody. I don't know if Nick, you've been collecting questions or are going to grill the panelists. Hopefully not me, because directing them at Martin, Christine, and Björn.
Tom, given that we're at the top of the hour, I think we'll need to conclude the webinar at this point. But I would like to thank you for moderating the excellent discussion. Like you say, we've covered a lot of ground, and I'd like to thank our panelists, Martin, Christine, and Björn. And thank you to the audience as well for joining the webinar today. I would like to apologise that we have experienced some technical issues today. An on-demand version of the webinar will be available in a few days. The Antibody Society will send a link via email to everyone who registered for the event. And please feel free to watch this or any of the Society's on-demand webinars when it's convenient to you. Thank you again to the panel and to the audience, and have a great rest of the day.
Thanks, everyone.
Thank you, everybody.
Thank you, everybody.
Thanks.
Bye-bye.
Bye.
Thank you.